All right, good evening. Thumbs up in the chat if you can hear me okay. Sound check, can you hear me? Fantastic. All right, my name is Hunter and this is Content Review for Principles of Biology students. Today we're going over the immune system. The first half of this is going to be just very definition heavy, review of some of the many components of the immune system that you should be aware of, and then we'll get into some kind of conceptual problems. All right, so bacterium and virus particles. These are simply types of pathogens. Helper T cell receptors and helper T cells. This is a type of immune cell that stimulates killer T cells, macrophages, and B cells to make an immune response. So this is a type that stimulates killer T cells, macrophages, and B cells. Cytotoxic T cells is a kind of T lymphocyte. Lymphocyte's just another word for white blood cell. These kill cancer cells in particular, or cells that are infected by intracellular pathogens, intra meaning inside, such as viruses or bacteria, or cells that are otherwise damaged. So lymphocyte, that is a white blood cell. B cells are kind of the star of the show. This is another type of lymphocyte that are responsible for humoral immunity component of the adaptive immune system. Adaptive immune system is one you'll hear a lot. You can kind of divide the immune system into those two broadest categories of innate and adaptive. Innate being the ones that are always active, and adaptive being the ones that emerge in response to specific pathogens. Component of the adaptive immune system. Yeah, so the B cells are the ones that are responsible for producing antibodies, which are a real key feature of the adaptive immune system. And memory helper T cells, we have memory helper T cells and memory B cells, and both are involved in the mechanisms of why vaccines work. So memory helper T cells are memory T cells are antigen experienced cells that mediate a faster and more potent response upon a repeat encounter with an antigen. Response. Similarly, B cells, memory B cells, are a type of B lymphocyte that forms part of the adaptive immune system. Lymphocyte is Memory B cells develop within germinal centers of the secondary lymphoid organs, and memory B cells circulate in the bloodstream in a quiescent state, sometimes for decades. So quiescent meaning inactive. Let's see. Cytokines. Cytokines are a lot of different substances, but these include interferon, interleukin, growth factors, which are secreted by specific cells of the immune system and have an effect on other cells. Um, many interleukin and growth factors. Interleukins, you might see this abbreviated IL. These are a type of cytokine first thought to be expressed by leukocytes alone, but we later found that they can be produced by many other body cells. CD4 protein's a cool one. CD4 cluster of differentiation 4. Cluster of differentiation number 4 is a glycoprotein, so it has a carbohydrate group attached to protein that serves as a co-receptor for the T cell receptor. Co-receptor. CD4 is found on the surface of immune cells, such as T helper cells, monocytes, macrophages, and dendritic cells. MHC, major histocompatibility complex. 1 and 2 molecules. 1 and 2 have slightly different roles that are related. These are also very important in the adaptive immune system. Class 1 major histocompatibility complex molecules span the membrane of almost every cell in an organism. So meanwhile, class 2 molecules are restricted to cells of the immune system called macrophages and lymphocytes. So pretty much every cell has class 1, and they usually present normal pieces from inside the cell, and so immune cells know that that cell is healthy and fine and just move on. When there is an intracellular invader, like a virus or bacteria, it presents pieces of them on these MHC complexes, and that is one mechanism that immune cells then know to attack that cell because it's infected. Antibodies, pretty straightforward one. This is a blood protein produced in response to and counteracting a specific antigen. Antibodies combine chemically with substances which the body recognizes as foreign, such as bacteria, viruses, foreign substances in the blood. Okay. Antigens are the toxins. Antigen is a foreign substance or a toxin which induces an immune response in the body, especially the production of antibodies. Epitope, epi, on top of, is the part of an antigen molecule to which an antibody attaches itself. Thymus. The thymus is a specialized primary lymphoid organ of the immune system, and within the thymus cells, thymus cell lymphocytes or T cells mature. Um, T cells are critical to the adaptive immune system, where the body adapts specifically to foreign invaders. We kind of talked about this, and the way to remember where T cells mature is the thymus. Starts with a T. So let's see. Primary lymphoid organ. Primary immune response being innate immune response. Uh, Bone marrow, we've kind of talked about before, is an important feature in the immune system because it's where all immune cells are made, right? Um, it's also the site of B cell maturation. So bone marrow is a spongy tissue inside some of your bones. This includes your hip and your thigh bone in particular. And there are many stem cells located in this tissue. Stem cells can develop into red blood cells that carry oxygen through your body, while white blood cells, these lymphocytes, fight infections. And we also produce platelets that help with blood clotting here. So, site of red blood cell, white blood cell, and platelet production. Hypothalamus is the homeostatic control center. This is a region of the forebrain below the thalamus, hypo, below thalamus, um, which coordinates both the autonomic nervous system and activity of the pituitary, controlling body temperature, thirst, hunger, and other homeostatic systems involved in sleep and emotional activity. Coordinates both the autonomic nervous system and pituitary activity. It's worthwhile spending a lot of time with the pituitary gland and learning all of its products, inputs and outputs, triggers for release, target organs, things like that. You will, if you continue in the Principles of Biology series, you'll have an opportunity to dive pretty deep into that. And again, if you do that in anatomy and physiology, um, which is actually where we are right now, if if you're taking anatomy and physiology concurrently. Let's see. Yeah. Fever is a temporary increase in body temperature. I'm sure most of you have experienced this. Um, this is often due to an illness, right? Having a fever is a sign that something out of the ordinary is going on in your body. Clonal expansion is when an antigen drives the proliferation and differentiation of cells with specific antigen receptors. An antigen drives the proliferation, so making more of them, and differentiation, specialization of cells with specific antigen receptors. Self versus non-self. This is kind of exactly what it sounds like. The key difference between self and non-self antigens is that antigens on the own body cells are known as self antigens, while antigens that do not originate in one's own body are called non-self antigens. So an antigen is any substance that induces immune system to produce antibodies. I'm actually going to put that at the top. Cool. So that was a lot of me typing. Um, we're going to do some more of that, but this part can be more conversational. I see a few of you in here. If you would like to engage with this, if not, if you're watching this in the future, pause, read these questions, move, kind of think through them, and then restart, and we'll kind of go through these. Um, what are pathogens? Why do we need to prevent them from colonizing our bodies? And if pathogens do manage to colonize, what effects can they have? Pathogen is a really generic term, right? This is any disease-causing organism. These include bacteria, viruses, fungi, protists. Um, our cells contain everything that is needed to support life. Sources of energy, um, a nice, watery, warm environment, right? These components support the lives of our own cells, but they can also easily support the lives of pathogens, which can then compete with our own cells and potentially destroy them. So what does this look like in real life? Um, when we get sick, when we have a pathogen in our body, they can do things like overgrow and put pressure on specific membranes or cavities. If you've ever had an ear infection, putting pressure on your tympanic membrane or your other spots in your inner ear, that can be really, really painful. It can obstruct your hearing. The structures of the inner ear are connected to much of your upper respiratory tract, and so that can spread and put pressure in your sinuses, um, tonsils, throat, lungs. If it gets really serious, your lungs can start to fill with fluid, which gets in the way of respiration and ventilation, like we were talking about in previous weeks, right? So we can literally have a buildup of foreign organisms so massive that it interferes with our tissue's ability to um, function normally. So overgrowth puts pressure on sensitive membranes or cavities. Eardrums can rupture, pressure builds and causes blockage and pain, things like that. These pathogens also produce toxins that can destroy our cells, right? They are competing for space and for resources, and one of the ways that they can do that is to kill the things around them. So, for example, some strains of bacteria produce exotoxins that destroy cellular structure. They can also parasitize cells and destroy their normal structure and function. Many fungal infections, for example, athlete's foot, are parasitic. Yeah, treating fungal infections is actually pretty tricky because they are more genetically similar to us than they are bacteria. For example, bacteria you can target cell walls, which eukaryotic cells don't have, um, which is part of the basis of why antibiotics work. Um, yeah, fungi are tricky. All right, what general defense mechanisms does the body use to help prevent colonization by pathogens? For example, what general defense mechanisms are involved in local inflammatory responses? So let's think first line of defense. Our skin is a protective organ, right? Mucose membranes in the nose and the mouth um, act as physical barriers to the entry of pathogens. Membranes. We also have glandular secretions that include lysozymes, keep our skin at an acidic pH somewhere between 3 and 5, which inhibits the growth of many microbes. So your sweat is literally antimicrobial. Um, one of many reasons to get regular exercise, I guess. Um, what general defense mechanisms are involved in local inflammatory responses? A localized injury, so a cut or a sliver, releases prostaglandins and histamines, which cause increased blood flow and swelling in the area. Let's write that part and we'll get to the rest of this. Prostaglandins and histamines. These are good vocab words to have, which cause increased blood flow and swelling. The clotting mechanism also helps prevent invasion um, by additional pathogens. So it's not just about stopping the loss of blood, which blood is energetically expensive to produce, right? Um, it is also creating a fibrous wall, basically, to prevent pathogen entry. We also have, and these are all part of the innate immune system. This is stuff that's active all of the time. Um, it is a really generalized response to an invasion or to an injury, right? Just kind of good to keep in the back of our mind. We also have phagocytic white blood cells. that move into the region and quickly consume pathogens that are present. And this is like a early line of defense to prevent colonization by harmful bacteria. Any time that you've had an injury that developed pus, those are the dead cell bodies of these phagocytic cells after they've done their job. Cool. So that was innate immunity. Now let's talk about or generalized immunity, general immunity. Now let's talk a little bit more about specific immunity, and in particular, B cells and T cells, and comparing them. So B cells, remember, are produced in bone marrow, as are T cells. B cells also mature in the bone marrow. Individual B cells respond to specific types of foreign antigens by secreting antibodies that interact with those antigens. So, individual B cells respond to specific types of foreign antigens by secreting antibodies. And these cause the cells to agglutinate, which is a fancy word for clump together. This agglutination tags or identifies the antigens for removal by phagocytic macrophages. Um, Phagocytic macrophages is like a level of specificity that I think is kind of funny. Phagocytic phagocytosis, right, is the mechanism by which it eats the pathogen. Macrophages, macro meaning big, phage meaning eat. It's a big cell that eats stuff by phagocytosing it. All right, basically engulfing it. Other antibodies can interfere with the function of antigens by binding with them and blocking their action. So you have two actions here related to B cells. You have uh, marking it for and causing this agglutination so that macrophages know to consume it, and then you can also, through that marking, have um, antibodies that interfere with the function of the antigens just by binding with them and rendering them non-functional. So comparing that with T cells, when body cells become infected with a pathogen, a piece of the foreign protein from the pathogen interacts with a MHC molecule, that major histocompatibility complex. And well, let's get that far first. Cell infection can occur by phagocytosis or by endocytosis. Um, and the MHC complex that involved is different between those two processes. The MHC antigen complex migrates to the surface of the cell's membrane, and individual T cells bearing complementary antigen receptors on their membrane surfaces interact with or bind with the antigen displayed on that MHC. So the MHC antigen complex migrates to the cell surface, and then an individual T cell, where an individual T cell with a corresponding Um, Sorry, I got reading the next sentence and totally lost my lost my train of thought. Um, Individual T cells bearing complementary complementary receptors interact or bind with the antigen displayed on the MHC. So the MHCs are like these little arms holding out these pieces of pathogen, like, hey, look at me, for T cells to come find them. Cytotoxic T cells, cytotoxic T cells bind to those foreign particles, foreign proteins rather, and the cytotoxic T cells act directly by killing the infected cell. Scorched earth policy. T cells respond by dividing and producing a clone of activated helper T cells and memory helper T cells. And the activated helper T cell produces cytokines, like we were talking about earlier, for example, that interleukin, which help the appropriate B cells to differentiate into antibody producing cells. And they also help cytotoxic T cells to become active in killing the infected cells. So there are lots of different subspecialized T cells that you should know about. We've already named a lot of them in this little bit here. So memory helper T cells and memory B cells are both involved in long-term um, accelerated response and immune memory. Differentiate into producing cells. Toxic T cells to become active. Cool. If about 10 to the fifth genes are available in the human genome to produce proteins, how can we produce more than 10 times 10 to the sixth different kinds of AB receptor proteins on B cells? I really like this question because it's kind of asking you to integrate a lot of what you've learned in previous, excuse me, in previous chapters and previous sections of your biology education. So I want to see if I have it. I don't. That's fine. The information in the question, the information that the immune system can produce more types of B cells than there are genes in the genome really begs this question, right? If each of the variable regions of the antibody on both light and heavy chains is made up of three parts and each is coded by a different region of the DNA segments for these three separate parts of each variable region undergo random recombination um, in the production of B cells. And as a result, many fewer genes are required to code for that 10 times 10 to the 6 different kind of AB receptor proteins on B cells. This is very similar to remember the codons and anticodons in DNA and mRNA synthesis, and how you know you have multiple multiple combinations that can code for the same um, polypeptides, and gives you some wobble, things like that. It's similar concept here. The variable regions and having these random recombinations gives you that, not infinite, but nearly adaptability to respond to ever evolving pathogens, right? How does HIV affect the immune system? I'm scrolling back up for a reason. Where is it? CD4 cluster of differentiation 4 is a glycoprotein that serves as a co-receptor for the T cell receptor. CD4 is found on the surface of T helper cells, monocytes, macrophages, and dendritic cells. This is a key aspect here. HIV targets cells that display both the CD4 receptor, so many of our immune cells, and a chemokine receptor. These two receptors are found on helper T cells. So infection by HIV ultimately leads to the death of helper T cells, and as a result destroys the immune response triggered by helper T cells. The adaptive immune response specifically. Consequently destroys the adaptive immune response responses triggered by helper T cells. So infected individuals, once HIV infection progresses to that point, infected individuals cannot fight off um, what otherwise would be really benign infections, and they may die. This is why some of the uh, not short-term, middle-term symptoms of HIV include things like ulcers in the mouth, because your body just stops being able to fight off some familiar bacteria um, or other pathogens that would otherwise not be a problem for someone with a healthier immune system. Yes. Yeah. Um, One of the reasons that HIV is really interesting from a clinical perspective too is because it's constantly evolving faster than we can really come up, faster than we could come up with vaccines, for example. So the parts of the virus key parts of the virus are not very highly conserved, the way that COVID-19 had really highly conserved aspects to its surface proteins that we could then replicate in the form of an mRNA vaccine, kind of hijacking our own cells' machinery to produce that part of the virus, which was not harmful to us, in order to develop this adaptive immune response. Um, HIV is hard because those parts are not highly conserved, and so any vaccine that we would make would not be useful for the next generation, which is evolving relatively, really quickly. However, um, study in that area led to far more effective antiretroviral treatments, which is now I believe the standard intervention for people infected with HIV. And I'm a little bit older, so I remember when we had no meaningful treatments for HIV and what a diagnosis could mean for people. And now it's very much like if you catch it early, you take antiretrovirals for the rest of your life, and it really may not even affect you. Most people with HIV walking around these days have undetectable levels of infection, which means that they are non-contagious. So just one of the many cool ways that medicine has progressed in a lifetime that I I think is just really cool. That's all I have for you this evening. Have a great weekend. Get some sun on your eyeballs. Enjoy the fact that the days are getting longer and longer. And I will see you all next week. Oh, we have a question in the chat. Would an infant born without a thymus be similarly affected by pathogens as those who are HIV positive? I love this question. This is a good conceptual question. And I'm only thinking about it for a moment because I don't have like a good clinical example like ready to go. But I would assume, since the thymus is the site of um, T cell maturation, that yes, that would result in an immature um, adaptive immune response. And I bet this is a Googleable question. Let's see. Result of congenital lack without a thymus. Congenital athymia. I'm going to just put this in the chat. Congenital, meaning you were born with it, right? Athymia, a prefix meaning without, thymia, without a thymus. May have profound immunodeficiency, vulnerability to potentially fatal infections, and life-threatening immune dysregulation. So yeah, that tracks. Are T cells not able to mature in the bone marrow like B cells? That is correct. So B cells again are creating those antibodies that trigger T cell response, right? So yeah, I don't know enough about the physiology of the thymus to say why that is. I just know that that's where that happens. Kind of like we know that the pancreas is where um, glucagon and insulin are made and released into the bloodstream. The thymus is the site of T cell maturation. Very different processes, but specialized organs. Cool. This is one of those topics that it's really worthwhile to spend some time with charts and images and start personifying these different cells and finding ways to remember their jobs. You are so welcome. Thank you for your attention, for your questions this week. I'm going to let you reclaim the rest of your evening. I'll see you all next week.